So today I will tell you how mitochondria are involved in both of these processes. Let's start off with an introduction, and then we have 5 parts that I will go through with you very stepwise, so I hope you will not be lost during this lecture. Let's start off with the introduction. Mitochondrial research, and namely redox-signaling, is a very active field of research, and it already started prior to 1900, so a good 100 years ago, with the discovery of mitochondria inside cells. Over the years 30s to 50s, much was learned about their function, for example during respiration and oxidative phosphorylation. And also, a major landmark was set with the discovery of DNA inside mitochondria during the 1960s. And this brought also a lot of attention to the fact that multi-subunit respiratory complexes are present in mitochondria, and then we learned more about their function, for example, during mitosis. Now, I already have to drink. I'm so sorry. Redox-signaling can easily be put into the scheme. Is it visible with the boards? So redox-signaling is easily fit into the scheme, and when you add the words mitochondria and redox in web of knowledge, you get around 3,800 results. For example, during biochemistry, cell biology, and even plant science. So it's really a topic of high interest. As you've heard in the lecture, the first lecture of today, metal pollution, but also diseases such as cancer, ischemia, hyperfusion, hypertension, and even a common cold can lead to a disturbance in the cellular redox state. So a disturbed balance between pro and antioxidant. And this is called oxidative stress. And this impacts the mitochondria to participate in a network of redox-signaling. When we look at redox-signaling, we have to keep in mind that all living organisms are redox systems. This means that they use anabolic or reductive processes to store energy, but they use catabolic or oxidative processes to release the energy. And redox signals are very important regulators of these processes, mainly during metabolism, but also morphology and development. And therefore, it's very important to control the balance between pro and antioxidants and keep it maintained, and therefore mitochondria can be involved. The function of mitochondria, I hope you all know, mitochondria produce ATP, so energy. They also produce ROS, or reactive oxygen species. They are involved in the uptake or release of calcium. They produce NADPH. They have their own DNA, so they make their own RNA and proteins. They are involved in thermogenesis, as you may remember, in brown fat, for example in hibernating animals or in small infants. And they are involved in the metabolism of amino acids, vitamins, and lipids, as well as in control of cell death. But ATP synthesis is their main function, and this granted them the name of cellular powerhouses. In tissues that have a high metabolic rate, such as muscles, the brain, heart, and liver, a lot of mitochondria are present. And this is how a mitochondria looks like. We have 4 well-defined compartments inside this organelle. First of all, we have an inner membrane, and this encloses a very folded space called the matrix. This is surrounded by an outer membrane, and then there is a space created between the outer and inner membrane that is conventionally called the intermembrane space. So we have 4 compartments, 2 membranes, an intermembrane space, and a matrix. In this inner membrane, proteins functioning in respiration are embedded, as you can see here. So this is the inner membrane. And we have 4 complexes, and sometimes also called complex 5, but mostly called ATPC. synthase that are involved in respiration and oxidative phosphorylation. Do you know the meaning of respiration? Does anybody know? Respiration? You cannot be wrong, I think. No answers? Really? Respiration, what do we do? Now I'm struggling with it. What do we need in respiration? Oxygen, indeed. And oxidative phosphorylation? Oxphos? What do we get? ATP, indeed. Higher phosphorylation. So I think I will have to explain it a bit more in detail, depending on your input. So, in short, electrons are fueled from complex 1 to 4, and ultimately, oxygen is reduced to water. So that's the respiration part. We respire oxygen. In the meantime, protons are exported into the intermembrane space and used by complex 5 to phosphorylate ADP to ATP. So in short, respiration is the reduction of oxygen, and oxidative phosphorylation is the ultimate production of ATP. So let's look at it more a bit in detail. At complex 1, we get an oxidation of NBH to NAD+, so we get electrons, and these are fueled with coenzyme Q. Complex 2 oxidizes succinate to fumarate, again delivering electrons to coenzyme Q. This electron carrier brings the electrons to complex 3, and there cytochrome C is reduced. This delivers the electrons to complex 4, and these 4 electrons are then used to reduce oxygen to water. So that's the electron transport part. In the meantime, complex 1, 3, and 4 public protons out of the matrix, and this generates a gradient that is then used to dissociate ATP. So I hope this is clear now to everybody because it's important for the next part of the lecture. Okay. So we know now a bit more about the mitochondria. How can they produce loss? The first lecture of today, I think you have seen this scheme before, so I will not go into too much detail, but you will remember from my first part that we need 4 electrons to get from oxygen to water in one time, the tetravalent reduction. But when only 1 electron is added to oxygen, we get a univalent reduction, leading to the production of superoxide. Again, 1 electron to hydrogen peroxide, and again 1 electron to the hydroxyl radical, which is a very toxic radical and very damaging. In the end, of course, 1 electron extra leads to the formation of water. When we look on a research timeline, between 1960 and 70, it was recorded that hydrogen peroxide was produced inside mitochondria. And also superoxide was detected a few years later. Also, they discovered that there were superoxide dismutase present in the matrix. Manganese superoxide dismutase able to reduce superoxide to hydrogen peroxide. And this brought a lot of attention to mitochondria as a source of loss. And nowadays, they are considered to be the quantitatively most important source of loss in most periodic cell types. Not all, as we will discuss later on. So, how are these raws then produced? This is again an overview scheme of the same mechanism that I discussed with you before. So electrons going through the electron transport chain, leading to the reduction of oxygen. And while complex 4 is very effective in retaining all electrons and all partially reduced intermediates of oxygen, the other complexes are not. And namely complex 1 and 3 are able to leak electrons to the matrix or the intermembrane space. And you can imagine there is a lot of oxygen present surrounding this electron transport chain because we need it to be reduced at complex 4. But when we have electrons escaping, we get the production of superoxide. When we get superoxide, it's fairly easy to get to other sources of frost, for example, hydrogen peroxide. In the literature, complex 2 is often described as a potential source of vacuum leakage, but this is still under discussion. So mainly 1 and 3 are the most important sources of frost. Now, you also heard in the first lecture of today that cells express a wide array of enzymatic and metabolic antioxidants during scavenge loss, and this is not different in the mitochondria. I told you before that superoxide is converted to hydrogen peroxide using superoxide dismutases, and they are both expressed in the matrix and the intermembrane space, and the only difference is the cofactor that they use. So in this case they use manganese, in this case copper or zinc, so they do the same. And the importance of this manganese superoxide dismutase is demonstrated by the fact that animals that lack this enzyme show perinatal vitality, so they will not survive. Compared to superoxide, hydrogen peroxide has a longer half-life, so it's more stable. It's able to cross membranes and therefore can act as a signal within the mitochondria or in the cytosol. But it can also interact with transition metals. I think you also have seen this in the first lecture today during the femto-metal-lyse reaction, where it can interact, for example, with iron and the superoxide to produce the highly toxic hydroxyl radicals. This will then lead to damage. So again, it's really important to neutralize this hydrogen peroxide. And in heart and liver cells, we have catalase being expressed in the matrix, which reduces hydrogen peroxide to water. In other cells, we have other mechanisms. For example, glutathione peroxidase. This is also expressed in the matrix. And it uses glutathione to reduce hydrogen peroxide to water. Of course, during this process, glutathione itself is being oxidized. So it needs to be back reduced to glutathione using glutathione dehydrogenase. You also have thioredoxins that do the same as the glutathione peroxidases. They get also oxidized during this process and need to be back reduced using a thioredoxin reductase. Both these enzymes depend on NADPH, and NADPH is a very important reducing equivalent in cells. And also, as I've seen when I talked about the function of mitochondria, you have seen that I said production of NADPH is very important. Now you see why, because this is needed for the scavenging hydrogen peroxide, because these enzymes need to be reduced for their cofactors. And mitochondria regenerate NADPH using different enzymes. For example, they have a transhydrogenase, which is an enzyme embedded in the membrane, and it uses protons from the electrochemical gradient to reduce NADPH. Or there are also enzymes present in the matrix that use TCA cycle intermediates, such as isocyclate or malate, to reduce NADPH. Either way, you can see the regeneration of NADPH costs energy, because here you use the proton gradient, and here you use energy supplying substrates. So scavenging of ROS requires energy. It's very important that you keep that in mind, that the ROS metabolism requires energy. Okay, so now we know that mitochondria produce ROS. How can they be measured? When we look at superoxide, I've told you before, it's very improvement due to the fact that the fluorescence is increasing. For hydrogen peroxide, other mechanisms exist, and the most commonly used is amplex red. It's a non-fluorescent dye, so under normal conditions there is no fluorescence. But again, when it comes into contact with hydrogen peroxide and also horseradish peroxidase, we get on one side a reduction of hydrogen peroxide to water, but a fluorescent product is formed. And here there is a direct relation between the amount of hydrogen peroxide and fluorescence. This is very reliable, sensitive, and reproduced. And why is that? We can directly quantify it due to the use of a calibration curve with known amounts of hydrogen peroxide, which is not possible with superoxide because it's immediately gone. Again, a research example using isolated mitochondria. Here you can see on the y-axis the fluorescence of amplex red and the time, and it's in isolated mitochondria. Over time, you see a bit of an increase of the amplex red fluorescence, and this means that under controlled conditions you get loss production. But this is also not only during stress; it's also under normal conditions that these enzymes leak electrons. But then, when you don't have stress, the balance is much easier to control. When you add antimycin A, does anybody know this compound? No? It's also called rotenone. Does that say anything to you? So rotenone is a complex 3 blocker of the mitochondrial electron transfer chain, which means that all electrons are building up here. They cannot go through to complex 4, and the percentage of leakage, the chance of leakage will be much higher because there are a lot of electrons present here. And you can see that we have an increased hydrogen peroxide production when antimycin A is added. On the other hand, when we add catalase, which is a scavenger of hydrogen peroxide, we can nicely see that it is decreasing. So this demonstrates that amplex red fluorescence is a direct proportional measurement of hydrogen peroxide production in mitochondria. Now, some people, some researchers directly use hydrogen peroxide as a measure for superoxide production. Because they say, okay, we have superoxide, it will dismutate to hydrogen peroxide, so when we know how much hydrogen peroxide is present, we know how much superoxide is present. But you have to pay attention because not all of the superoxide is converted to hydrogen peroxide. There are also other components present in mitochondria with which superoxide can interact, and one of them is nitric oxide, and this can form two peroxynitrites in contact with superoxide. In addition, hydrogen peroxide is not only produced in the matrix, it's also produced in the intermembrane space and in the outer membrane, so it does not only reflect the matrix production of superoxide. And not all of the matrix hydrogen peroxide will survive its effluence from the mitochondria because enzymes of the antioxidant defense system will reduce it. So you can use hydrogen peroxide production as a measure for superoxide production, but you need to add superoxide sinks, such as nitric oxide. You need to add the fact that it is degraded, and you need to subtract the amount that is produced outside of the matrix. Okay, so what are produced? We know we can measure them. What is the consequence of this loss production? And the definition of oxidative stress, I think you also heard before today, is a disrupted balance between pro and antioxidants in favor of the former. In mitochondria, this is a little bit extended to the fact that electron transfer reactions are disrupted, leading to this imbalance. And this can either lead to damage when ROS levels are very high, and it underlies several pathologies, such as ischemia, but also it can lead to signaling, and then we call it retrograde signaling. Retrograde signaling means signaling from organelles to the nucleus, and not the other way around, like it's normally done. The nucleus gives a signal to the organelles. That is called anterograde function depending on the stress intensity and duration. How is damage occurring? Let's look at the scheme. We know that the respiratory chain produces loss due to electron leakage. This loss can then oxidatively damage proteins in the respiratory chain. And when you have damaged proteins, it's easy to imagine that there is more electron leakage occurring, so this is actually the vicious circle. On the other way, they can also damage the mitochondrial DNA, that's very close to the respiratory chain. And mutations and deletions will again lead to the build-in of defective proteins into the respiratory chain. Again, we get the vicious circle. Last but not least, lipid peroxidation can occur, and there can be holes occurring in the membranes of the mitochondria. And when this occurs, cytochrome C can leave the mitochondria and can induce apoptosis. Also, the inner membrane can get coarse, and this is called permeability transition pore. Here it's the outer membrane polarization. So we get a dysfunction of our mitochondria because of the respiratory chain that is dysfunctional. We can get apoptosis and necrosis. They can interact with each other and lead to disease. Mitochondrial DNA, it's a double-stranded DNA molecule, and it encodes proteins of the respiratory chain, as well as transfer and ribosomal RNA. And it's very sensitive to oxidative stress, more sensitive than the nuclear genome. And there are different reasons for this. One of them being very obvious. We only have one nucleus, or one nuclear genome per cell, but you can get up to 1,000 copies of mitochondrial DNA, depending on which tissue you're in, of course. In heart cells, this can actually be the case. So we get more copies, so more chance of damage. It's also very close to the respiratory chain, so very easy to be attacked by ROS. And it's also easily accessible because it has no histones, it has no introns, and also no DNA-associated proteins as compared to the nuclear genome. And last but not least, DNA repair exists, but is very limited in mitochondria, which means that once damage has occurred, it's not easy to recover from it. Then the involvement of mitochondria in cell death. We first have to describe the two types of cell death that can occur, and I think you know that necrosis and apoptosis are their most important forms. And this table just lists the big differences between the two. And induction of this type of cell death is very different. For necrosis, it can be injury of the tissue, while for apoptosis, specific signals are required. And that's why we also call it programmed cell death, because it's really a signaling process occurring inside a cell. And another big difference is the fact that while ATP is lost during necrosis, it's required to perform apoptosis. Mitochondria are involved in both types of cell death, although the induction stimulus is different, because the permeability of the mitochondrial membrane will in both cases be increased. For necrosis, this will ultimately result in the formation of flaps and the rupture of cells, meaning that the cell is completely disappearing. And ROS can, of course, enhance this permeability increase, as we have seen in the previous scheme. In apoptosis, the outcome is different, but the effect is the same. So, you get a release of cytochrome C and the activation of caspases in the intrinsic apoptotic pathway. And again, ROS can be involved in this process, but also some of the pro- and anti-apoptotic proteins, such as RAX and BCL2. Let's return back to the research with an example. Again, the ischemia reperfusion of the heart cells. And researchers want to demonstrate the relation between ROS and cell death induced by mitochondria. How did they do this? Well, first of all, they used clopidogrel iodide. I don't know if anybody knows this dye. What does it do? Anybody knows? PI? Iodide. What does it do? For necrosis. Why is it forming process or cell death in general? It's a dye that is only being taken up by dead cells. So it colors actually the dead cells. And here you can see a baseline condition cells, and here you can see cells that suffer from ischemia or infusion. So we get an increase in PI uptake. So this means cell death is occurring during ischemia. Okay, but what kind of cell death? Therefore, the researchers used mitochondria, and this dye colors mitochondria. As you can see, a lot of red dots are visible. There are heart cells, so there are a lot of mitochondria. And they also use cytochrome C fluorescent probes. And cytochrome C, normally, in normal conditions, is present inside the mitochondria. But here, they merged both pictures and saw that the green color was mainly outside of the mitochondria. And this loss of colocalization means that apoptosis is occurring. As we have seen in the scheme, cytochrome C has to leave the mitochondria for apoptosis to be induced. So we have cell death, and we know it's apoptosis. Are ROS involved in this process? Therefore, the researchers overexpressed different antioxidative enzymes in these cells, and they saw that when they overexpressed here the black bar, which is SLE2 or the manganese superoxide dismutase, you see less cell death. So less cell death when we overexpress manganese superoxide dismutase, meaning that superoxide has to do something with the induction of cell death. And yet another way to tell the same story, they used mitochondrial row GFP, and this is a probe that's not only targeted to mitochondria, but it also reflects the oxidation state of the mitochondria. So when fluorescence or the oxidation of the probe is increasing, it means that the mitochondria are suffering from oxidative stress. And here you can see for ischemia, you get an increase of the oxidation of the probe, which means mitochondrial oxidative stress is present. When you add NEC, here, this line, you see a clear decrease of the oxidation state of the probe and less mitochondrial oxidative stress. So, ROS production from the mitochondria is involved in the cell death induction during ischemia infusion. Okay, when levels of ROS are lower and mild oxidative stress is occurring, you can also get signaling mechanisms. And the main question here is how can ROS translate into redox signals? And one of the main mechanisms is the fact that we have proteins in our cells, and these proteins contain cysteine residues, and they contain diol groups. As you can see here, this SH is a diol group on the surface of a protein, and this can serve as a cellular redox sensor, meaning that it can be either reduced or oxidized, depending on the amount of ROS that are present inside the cell. And proteins expressed in the mitochondria are very rich in cysteines, so very good targets for this redox modification. Three of the main mechanisms to modify proteins, redox modified proteins, are glutathione, nitrosylation, and the formation of disulfide bonds, which we will discuss in a bit more detail in the next slides. But it's very important to keep in mind that this modification is reversible, which means that you can give a signal, but you can also stop the signal, depending on what state you're in. And how can the activity of the protein then change during a modification? Well, you can easily imagine when this diol group is present in the active site of the protein and it's modified, the activity will change. And in addition, the conformational change can also be induced, which of course will also alter the activity of the protein. The first mechanism, S-glutathione. Does anybody know why this S is there?
What is okay?
Sorry?
What is okay?
It's just because of the diol group, and SH is a diol group, so that's why the S is there. But you can also just call it glutathione. It's the same. So here, the interaction between the diol group and an oxidized molecule of glutathione leads to a modification of the protein with the oxidized glutathione attached to it. it's a really reversible modification. Nitrosylation, as the word says itself, it's an interaction with nitric oxide. And here you can see that there is an N to the protein. Again, the antioxidative defense system can modify this, can reverse this modification. And then the formation of disulfide bridges. I think it's the most easy to understand. You can get two chiral groups interacting within the same protein, and then you have an intramolecular disulfide bond. Or you can get two proteins with two chiral groups interacting, and you have an intermolecular cysteine bond. And then it's easy to imagine that these two proteins will not be able to function as well as they did before. Again, antioxidative defense can be used to reverse the circle. So, redox signaling via these mechanisms can occur within the mitochondria on proteins that are expressed in the mitochondria themselves, but it can also communicate to other compartments of the cell. For example, when kinases are modified, transcription factors that go to the nucleus can also be modified, ion channels can be modified, and apoptotic proteins. This is again a scheme to simplify the story a bit more. So we get ROS production, for example, hydrogen peroxide at the respiratory chain, and the enzyme activity in mitochondria can be altered. But hydrogen peroxide can also leave mitochondria and then signal to the cytosol, for example, by changing cytosol enzymes. It can indicate something about the mitochondria. So mitochondria signal, okay, I have a problem, do something with it. And the cytosol gives back the response, for example, due post-translational modification or an altered supply of substrates to the mitochondria. There can also be signaling to the nucleus via redox-sensitive transcription factors. And this is more a long-term response because it requires synthesis of new proteins coming from the nucleus to the mitochondria. And then you can also have just the production and the signaling. Not only hydrogen peroxide itself can lead to signals, which we call primary redox signaling. It can also lead to damage to lipids, for example, and this can then lead to secondary redox signaling because it's not a direct loss-related effect, but an indirect loss-related signaling. Now, we know that mitochondrial oxidative stress is underlying several diseases, so it's often used in therapy. And here it's important to keep in mind that mitochondria are both important for life as well as for death, because they produce ATP, but are also able to reduce apoptosis. So depending on what you want to target, you have to be very careful. For example, cancer cells, they require a lot of ATP. So when you want to target cancer cells, you have to target the mitochondria by inhibiting them. You have to disturb respiration and glycolysis so that they cannot respire and produce ATP anymore. But when you have oxidative stress underlying your disease, for example during ischemia, you do not want to kill your cells, you want to rescue them, and then you have to use antioxidative drugs. And there are different ways to target these drugs to mitochondria. You can make use of their biophysical properties, meaning you have a very low membrane potential, -180, and when you have a positively charged molecule or drug, it will be selectively targeted to mitochondria because of its positive charge. In addition, you can use a prodrug that is only being activated by a mitochondrial enzyme and not by any other enzyme, so it will only be active inside the mitochondria. And thirdly, you can use a specific transporter that's only present in mitochondria so that the drug will only be delivered in the mitochondria. Of course, when you target your ROS production, you have to keep in mind that they're also involved in signaling. So again, the balance between damage and signaling have to be respected. So it's very important to keep in mind. And during clinical trials, it's always the same that they look at effects of antioxidative drugs and see effects, side effects, because they disturb also signaling. A proof of principle, so that you can believe that this really works. that the color is disappearing after 34 minutes. When we add mitoGSH, which is a glutathione proline ester, so a really therapeutic drug, we can see a delay in this depolarization. So we get it only after 54 minutes. It's still happening, but it's delayed, about 20 minutes. And the mechanism that is used here to target the mitochondria, it's a positive charge of this group on the molecule. Another example, EPPP, this compound. It has the antioxidant capacity of vitamin E, and again, it has a very positive charge, so it will be selectively brought into the mitochondria. And we can see, depending on the concentration, that the oxidative damage to either lipids or proteins will decrease. So again, we get an antioxidant to the mitochondria, and we get not only less loss, but also less damage. And that's of course what we want. Okay, I'm going very fast. I hope that's good for you. I hope you can follow, because otherwise, please disturb me. Now I come a bit with my research. I do research on plants, and in these organisms we have some special enzymes that are not present in animals. And I want to tell you about their benefits and how we can exploit them in mitochondrial oxidative stress in animals. So this is again an overview of the respiratory chain, as we have seen before. And in plants, we have some additional enzymes, such as AOAs, or the alternative oxidase. We also have alternative NADPH dehydrogenases, and these are located on the external or the internal side of the inner membrane. And we have uncoupling proteins. And here I cheated a bit, because I think most of you know that uncoupling proteins are also present in animal cells. But I swear AOAs and NADs are not, so they are plant specific. How do these enzymes function? These enzymes oxidize either cytosol or matrix, NADH or NADPH, and deliver the electrons to coenzyme Q, which directly delivers the electrons to AOH. And then AOH does the same as complex 4 is doing. It reduces oxygen to water. But it's a shortcut, as you can see at the beginning of the electron transport chain. So less electrons are able to escape and less ROS are produced. Uncoupling proteins function a little bit different. They dissipate the proton electrochemical gradient. They transfer protons back, as does the ATP synthase. But the big difference is they do not produce ATP. So you get an uncoupling of respiration and oxidative phosphorylation. These enzymes in plants are very important in maintaining the redox balance and also sustaining the mitochondrial metabolism, as we will see in the next few slides. Let's start off with the alternative oxidase. This is a picture of tobacco cells, so tobacco plants cells. And we have two dyes, DCF and MT. DCF, does anybody know what this is? It's a ROS dye, so it colors ROS, for example, hydrogen peroxide. And MT is mitotracker, so it colors mitochondria. These are wild type tobacco cells, so nothing changed to them. These are cells AS8 that have a lower AOX expression, and S11 are cells that have a higher AOX expression. And as you can see, under control conditions, we have no ROS production in our wild type, no ROS production in our cells that have a higher AOX, but we have a lot of ROS production in the cells that lack or have a lower AOX expression. And you can see, when you imaginally merge both pictures, that they are mitochondrial. So less AOX equals more ROS. Then we challenge the cells, and we give them antimucin A. As you remember, this is an inhibitor of complex 3, so we get more ROS production. As you can see here in the wild type, nicely co-localizing into mitochondria. But a massive increase of ROS production when we lack AOX. And in cells that have a higher AOX, you do not see anything. So again, less AOX equals more ROS, and this means that AOX is able to lower mitochondrial ROS production in plant cells. So, because of the fact that they discovered this in plants, some brilliant people thought, what if we use AOX in animals to counteract diseases where we have mitochondrial oxidative stress or where we have oxalate deficiency? And this led to a first proposal to use this AOX, and also people that constitute the alternative consortium. They were able to successfully express AOX in human cells, even rescue COX-deficient human cells and flies, and they were able to express AOX in mice. But this is where the research stops for now, so they were not able to do more with this. Still, they think that AOX is very promising to use as a gene therapy to tackle oxidative stress in humans, based on the story of plants. What about the uncoupling proteins? In plants, superoxide production stimulates their activity, and when they are overexpressed, you get an increased tolerance to oxidative stress. So again, people thought, hey, what if we use these uncoupling proteins to counteract mitochondrial oxidative stress? And already 10 years ago, there was a paper published in Nature by Ete and colleagues, and they saw that when you have an increased superoxide production, you have an activation of uncoupling protein 2, which will increase the permeability of the membrane, because of course protons are flowing back. This will reduce the membrane potential and inhibit further superoxide production. So yes, the same mechanism occurs in animal cells as in plant cells. So they thought, okay, we can use uncoupling proteins. But this is also where the story ends. So I think there is a lot of research that still needs to be conducted to exploit their benefits. Okay, this already brings me to the final part about my PhD research. So what I'm studying in our model organism Redox-signaling, which you can see here, is the relation between metal-induced oxidative stress and plant mitochondria. So, as you have heard today, industrial and agricultural activities have led to a persistent metal pollution in our environment, and specifically here in the northern Campine region of Belgium, and also a little bit in the Netherlands, we have the cadmium pollution problem. Because these smelters in the last century emitted also a lot of cadmium to the environment. Cadmium is a non-essential metal, and it's also non-redox active, but it is able to disturb the balance between pro and antioxidants. And what we are studying is this balance, and also how it can lead to either damage or signaling and inflammation in the modeling organism amyloidosis right now. This is an overview of the research questions that we asked to ourselves in the beginning of my PhD. So we knew that cadmium exposure can alter the redox state of cells, but we thought also that it might impact the mitochondria, leading to retrograde syndrome and alternative respiration activation, which could then maybe limit ROS production and lead to acclimation of the plants to cadmium exposure. So a bit more in the working model, we think that metals can enter plant cells, can enter the mitochondria, and disturb the balance between ROS production and antioxidant defense, because they can lead to a dysfunctional electron transfer chain, overreduction of the chain, and thus more ROS production and thus damage. However, we are not sure that cadmium or metals in general are able to enter mitochondria, but we do know that they enter the cytosol, and once they are present inside cells, they can interact with all cellular compartments. think that wells, for example, may signal to the nucleus, hey, we have a problem here in the mitochondria, and then there is an activation of AOX, and potentially also alternative NDPAs to that protein or encoupling proteins to minimize this loss production and to restore the balance again. So our hypothesis is that the alternative respiratory pathway ameliorates cadmium-induced oxidative stress. How did we study this hypothesis? We used Redox-signaling, a very small plant, as you can see here, and grew them in hydroponics under LED lights, as you can see here. And hydroponics means that the plants are facing with the leaves to the light, and with the roots, they are in black boxes, so the roots get no light, as it is in nature, of course. And after 20 days of growth in hydroponics, we added cadmium to the nutrient solution, and we used either 5 or 10 micromoles. And these concentrations are actually similar to the concentrations found in our environment, so they are environmentally realistic and sublethal, so the plants survive, because otherwise we cannot study anything of those. And then we sampled roots and leaves at just before the exposure started, and at regular time points after the exposure started, so that we were able to really study the sequence of signaling events and responses happening inside the plants. What did we study? Yes?
Why don't you expose the plants from the beginning? Because you expose them now.
That's true. So why do we expose them only after 20 days? Because, so it's right, in the environment, the metals are present and the plants are growing on the metals. But here we want to study a mechanism. And then when you give cadmium from the start, this mechanism will already be active in very small plants. So you can imagine to study, for example, gene expression in these plants will be very difficult. I don't know if you have seen sometimes little small boxes. So it's only like this high. We cannot get enough plant material to study responses in young plants. Because then we have to study in other ways. But here we chose to expose them and to study the induction of a mechanism when you give cells. But it's pretty hard, because that's another setup that can also give a lot of information. Okay, so first we took a look at the alternative oxidase, which, as you remember, is able to directly reduce oxygen to water. And in plants, we have a gene family composed of 5 members. 4 of them belong to the AOX1 group, and we only have 1 AOX2 gene. And the difference between the 2 groups is that AOX1 is presently boost, while AOX2 is always present. So we only took a look at the AOX1 genes because we added cadmium. And the 4 isoforms here are differentially expressed in different organs, with AOX1A known as the most dominant isoform. We checked their expression using real-time PCR and cyber-glue technology. I hope you're all familiar with this technique. I think so. And also verified this gene expression at the protein level using Western blotting and specific antibodies or AOAs. These are the results for the roots. On the y-axis, AOX1 isoform expression relative to the control at 0 hours. And on the x-axis, you can see at the different time points the expression in plants that are not exposed or exposed to either one of the concentrations. And as you can see, from 24 hours, we get an induction of AOX1A expression, while 1C is not visible. So in the roots, AOX1A is the most dominant isoform, and it is activated at the transcript level by cadmium exposure. On the protein level, we were also able to detect the difference, as you can see here, with bigger bands, after 2 hours for 5 and 10 micromoles of cadmium. But after longer time points, there was no change in protein, although gene expression level was still increased. In the leaves, the story was a little bit different, because AOX1A was also increasing during cadmium exposure, but another isoform popped up, the 1B, and you can is not present. So during cadmium stress, there is an extra isoform that is being induced in the leaves. And again, we were able to demonstrate an increased protein AOX level via Western blotting. So AOX is induced both at the transcript and protein level by cadmium exposure. Then we took a look at the alternative NADPH dehydrogenases, or the NADs. And as you remember, we have both NADs on the internal as well as on the external side of the membrane. A and C are the ones at the internal side, and the B group are the genes that are present on the outside, the external side. Again, we measured their expression using the real-time PCR, and these are the results for the groups. So ND isoform expression, again the same scale on the x-axis. And as you can see, the most important change is this purple one, and it's NDP2, so an external isoform that is inclusion. When you look in the leaves, it's even more obvious. So we get blue isoforms, NDA1 and C1, both internal isoforms under control conditions, and we get the purple isoform very present in cadmium stress conditions. So we think there is a shift occurring, mainly in the leaves, from internal to external isoforms being active. And on the scheme, you can see that they can then function via the cytosol, use the electrons, and deliver them to AOX to function as an alternative chain during cadmium exposure. To conclude the story, we think that, and we know, that cadmium activates alternative phosphorylation mediated by AOX and NDs at the transport and protein level. We know that AOX could be a very important mediator of cadmium-induced responses, and that they can co-function with external NDs during metal stress to function as a complete alternative chain for the dysfunctional electron transfer chain. In future experiments, I will discover more about the function of this dominant AOX1A isoform using either plants that overexpress it or lack AOX1A. We will discover more about their cadmium sensitivity, because it could be that without AOX they are more sensitive to cadmium, or with more AOX less sensitive. And looking at the response of other alternative components can also shed more light into these processes. This was my story for today. Thank you very much for your attention.